Hi guys, so today we will be solving the paper for NEET 2024. Okay, a pretty easy paper, I must say. There were direct questions, nothing much to think about. While we will solve this paper, we will also look at methods of elimination, how to easily do match the followings and all of that. Yeah, let's start. I'll read the question and I'll solve it for you. Okay, and wherever required, I'll give you explanations as well. Let's begin. So section A, as you know, NEET now has differentiated its questions into section A and section B, right? Section A has 35 compulsory questions. Section B has 15 questions, out of which you have to do 10. 5 would be optional, a total of 45. This is true for all sections. So we will start with section A of Botany. Question 101 for my particular booklet. Identify the set of correct statements. So these are called the statement type questions, right? So you are given a number of correct or a number of jumbled statements. There are multiple formats beyond which these questions can come. So they can ask you to find out which of these statements out of the 5 are correct, which of them are incorrect, what is the correct order. If it is a process, then what is the correct order of the process can be. So these are the multiple formats in which this question can come. So let's start with the first one. Identify the set of correct statements. Statement A is, the flowers of Vallisneria are colorful and produce nectar. Vallisneria is an aquatic plant, right, which is very uniquely wind pollinated. And as you know, wind pollinated flowers which are not pollinated by insects or animals that consume the nectar or are attracted by the brightly colored petals of the flower, are usually pale colored or white. They do not have any strong smell and they do not have nectar. So the first statement where it says that the flowers are colorful and produce nectar, it's obviously wrong. So A is wrong. Now let's look at the options. In option number 1 and in option number 4, A has been included, which we know is a wrong statement. So these two options, 1 and 4, are obviously eliminated, right? So we have to pick from 2 and 3. Now let's read the second one. The flowers of water lily are not pollinated by water. True, it is pollinated by wind. So this is a correct statement. Now out of options 2 and 3, only option 2 has enlisted the statement B as a correct statement. So obviously option 2 is our correct option. When you follow the method of elimination, it saves you a lot of time, because now we don't have to read C, D and E and think whether they are correct or wrong, because we obviously could eliminate options 1 and 4 based on A, and we could select option 2 on the basis of B, right? So correct option for this question would be option 2. Question 102. The type of conservation in which the threatened species are taken out from their natural habitat and placed in a special setting where they can be protected are given special care is called. So now let's see what conservation is. So if this is the habitat, which is the original habitat for a species, and we choose to conserve it in this habitat itself, then it's called in situ conservation. But if we take it from its original habitat to a different location offering the same niche, then it would be an ex situ conservation. So here the question is obviously talking about ex situ conservation, because the species has been moved from its original location. Let's look at the options. Semi-conservative method. Now this is a funny option, because this option obviously is not a part of conservation, it is of DNA replication, right? Sustainable development, which is option 2, means to do development in such a way such that our future generations are also able to enjoy the perks of the nature that we do. So to do development in a sustainable fashion so that we do not over exploit or exhaust our resources. In situ conservation obviously is not the case that is being talked about. So the last option that we are left with is biodiversity conservation. This is biodiversity conservation. Obviously, it's the umbrella term. Now, question 103. Inhibition of succinate dehydrogenase enzyme by malonate is an example of. So it is really a classical example of competitive inhibition. If you would refer to the videos of enzyme kinetics, you would be able to understand the different types of competition that we have talked about there. Moving on to question 104. Identify the part of the seed from the given figure. So they have given you the diagram for monocot seed. This is specifically a kernel of a corn, right? So in this question, question 104, A obviously is the coleoptile, which protects the plumule. B is the plumule. C is the radical. And D is the coleorhiza. So we know that the plumule gives rise to the shoot system and the radical gives rise to the root system. So the question asks for the part which is destined to become the root. This is option C. Radical, which is the correct option, that is option A, 1, that is. Okay. 105, another straight question. Bulliform cells are responsible for. Bulliform cells are found in monocot leaves where they, by the virtue of turgidity and flaccidity, they prevent the inward curling of leaves in monocot. So the correct option is option C. 106. Which of the following are required for the dark reaction of photosynthesis? The dark reaction of photosynthesis is also known as the light independent reaction, which basically means that this reaction does not require what? It does not require the presence of light. It can happen during daytime, but it does not require sunlight. So obviously light is not required, and light in plants is absorbed by chlorophyll. So yeah, chlorophyll is also not specifically required for this reaction, am I right? Now, however, this reaction is also where CO2 fixation takes place, right, but via the C3 or the C4 cycle. So therefore CO2 is required, and the ATP, which is produced by photophosphorylation, and NADPH are also required. So C, D, E are required, which comes under option 1. So that's your correct option. Moving on to 107. Formation of interfascicular cambium from fully developed parenchyma cells is an example for. So now, in question 107, we are talking about what? We are talking about differentiation and de-differentiation. So a meristematic cell is a cell which is dividing always in plants. It attributes to plant growth. However, it does not have specialized functions. When they attain specialized functions, they become permanent cells. Right? This process is known as differentiation. So differentiation is the assignment of a specific task or a function to a particular cell group, right? So now, parenchyma cells actually then get de-differentiated to form the cambium during secondary growth. So they first change into meristem and then they change into cambium. This happens during secondary growth and it is de-differentiation, because they will first change into meristem-like cells. So whatever special ability they had attained, they are going to lose, right? So therefore they are de-differentiated and then they change into cambium. So the correct option is again option 1. Moving on to 108. Hint 2 always cuts DNA molecules at a particular point called the recognition sequence, right? So now let's talk about our restriction endonucleases. So what are restriction endonucleases? They are nucleases. So enzymes that are able to cut the DNA but at specific sites. Now restriction endonucleases have specific recognition sites. So for this particular example that you have, hint 2, the recognition site is a 6 base pair recognition site, which is option 4. That is your correct option. Okay. Now, moving on to question 109. Tropical regions show greatest level of species richness because. Okay, so as we know that tropical regions closer to the equator, species richness is higher, and the more you move towards the pole, it dwindles, right? So let's look at the statements. Tropical latitudes have remained selectively undisturbed for millions of years, hence more time was available for species diversification. This is a correct point. Option B says tropical environments are more seasonal. No, right? This is wrong. So let's look at the options where B is entailed. So 1, 2 and 4 have B in them, and B we know is wrong. So therefore the correct option is option 3, A, C, D, E only. Moving on to the next one, 110. Which one of the following is not a criterion for classification of fungi? So when 5 kingdom classification was done, it was initially done on the basis of cellular organization. So either the cells were prokaryotic, they were all included under Monera, or eukaryotes, right? These eukaryotes were then classified on the basis of number of cells. So unicellular eukaryotes were put under Protista, and multicellulars were then further classified on the basis of the presence and absence of the cell wall. So if cell wall was absent, they were animals, and if the cell wall was present, then again they were classified on the basis of heterotrophy or autotrophy, and the heterotrophs were called the fungi. So fungi was primary identification for fungi was multicellular eukaryotic organisms with cell wall that are heterotrophic in nature. That is what was the major point of differentiation from plants, right? And therefore the correct option would obviously be fourth one, the mode of nutrition, which is heterotrophy. Now moving on to question 111, another direct question. How many molecules of ATP and NADPH are required for every molecule of CO2 to be fixed by the Calvin cycle, right? So that is the C3 cycle. It requires 3 molecules of ATP and 2 molecules of NADPH. So the correct option will obviously be option 2. Moving on to 112. These are regarded as major causes of biodiversity loss. You have to choose the correct ones. Now, the reasons for biodiversity loss are enlisted in 4, and they are known as the evil quartet. The evil quartet consists of overexploitation, habitat loss and fragmentation, co-extinction, and the danger from invasive species. Out of these, 3 are here: overexploitation, co-extinction, and habitat loss and fragmentation, A, B, and D. So that's your correct option, option 2. Question 113. The capacity to generate a whole plant from any cell of the plant is called. So now this is the concept of stem cell, right? So for question 113, you have to understand what a stem cell is. Very easily speaking, a stem cell is able to differentiate into any cell depending on the potency, while also regenerating the stem cell reservoir, right? So they will basically, when they will do mitosis, they will divide one to give you a new form of cell, let's say cell A, and the other daughter cell is going to replenish the reservoir. So what then is the potency? Potency is the ability to differentiate into a diverse group of cells, right? So based on that, potency can be total, plural, multiple, or uni. That is one. So total matlab one cell, for example, the zygote that is formed after fertilization, we were also zygotes. And then from that zygote, everything that we have in this adult body now, from our blood to our internal organs, to our cardiac cells, to our Purkinje fibers, to our nerves, everything was formed from these cells. So then it becomes totally potent, which is totipotent cells. If a cell has multiple, so germ cells, for example, your ectoderm, endoderm, and mesoderm, they are pluripotent cells because they can give rise to multiple types of cells. For example, your ectoderm has formed your skin, your bones, as well as, not bones, your skin, your nervous system, all of that is of ectodermal origin. And multiple would be multipotent. So for example, hemopoietic cells can give rise to any of the blood components, right? And unipotent is ones that can give rise to only one kind of cell. So your germ cells, primordial germ cells, are unipotent. Okay, so in this question they talk about generation of a whole plant from a single cell. That is only possible from the zygote, which is a totipotent cell. Then the potency becomes totipotency. Okay, moving on to question 114. This is the equation for the Verhel's-Pearl logistic growth. So logistic growth talks about, see, initially when we talk about population growth, it is obvious that in a given scenario, population can grow exponentially, right, geometrically. But based on a theory given by a very famous man known as the Malthusian theory, he said that resources have an arithmetic progression, whereas life has a geometric progression. So for any given ecosystem, while the resources within the ecosystem are multiplying themselves arithmetically, life in that ecosystem is multiplying geometrically. Therefore, there will come a time when growth will become stagnant. This is the logistic growth curve, right? It's a sigmoid curve which says that after a certain point, the population will plateau. This is the plateau, right? So it is differentiated into a lag phase, into a log phase, and into a plateau, right? So in this, why is the population reaching a plateau? Because every ecosystem has something which is known as its carrying capacity, its maximum ability to support a certain number of organisms of different species, obviously based on the resources, availability of resources, right? So this carrying capacity is referred to as K, which is taken into consideration in the logistic growth curve equation. So K stands for the carrying capacity. Moving on to 115, spindle fibers. Now, spindle fibers are produced during cell division from the centriole in plants and from the from the centriole in animals and from the microtubules in plants, if I'm not right. So, therefore, when does it happen? So, in the prophase, obviously the cell dissolves, the nuclear membrane, the centrioles divide itself, the chromatin will condense into chromosomes, and then the metaphase will begin, where the spindles will come and attach the centromere, which contains the kinetochore, and start the process of pulling it apart in anaphase. And in telophase, the reversal of prophase happens. So the correct option here, spindle fibers attach to the kinetochores of chromosomes during the metaphase. The fourth option is the correct option. Moving on to 116. Identify the type of flowers based on the position of the calyx, corolla and the androecium with respect to the ovary. So if the position is, it is surrounding the ovary, then it is called perigynous. If it is below, it's called hypogynous. If it is above, it's called epigynous. Very clearly in both the diagrams here, you have perigynous flowers. So the correct option will be option 2. Moving on to match the following. Let's look at the trick in match the following. Okay. So for match the following, you look at the first column and try to figure out the ones that you instantly know, that instantly click. Like for example here, for me 2, Rhizopus and Agaricus are clicking instantly. I know Rhizopus is a bread mold, so that's 3. And I know Agaricus is a mushroom, which is 1. So I will look for A3, D1 options. If I look for A3, D1 options, there is only one of them, which is option 3, then I know this is correct. I don't have to be bothered about B and C, right? So option 3 is correct for this one, A3, B2, C4 and D1, okay? Next one, genetics, right? So 118, let's solve this question on the board. It says that a black seed color plant, it could be capital B or capital B small b. So then the phenotype of a black seed is the dominant phenotype is crossed with a white seed, which is the recessive factor. Yeah. So in order to find out the genotype of the black seed plant, which of the following genotypes will you cross it with? So they have said that this is dominant, this is recessive. To figure out which one of them is the correct genotype of your specimen, what will you cross it with? This is a classic question of a test cross. So what is a test cross? A test cross is when we take a dominant phenotype organism and cross it with their recessive parent. What it does is very clear. Now, in this case, we have 2 possible outcomes. Case 1 would be if it is capital B homozygous crossed with small b, then all the progeny would be what? Then all the progeny would be heterozygous dominant, which means they will all be black. However, if my organism is like this, then let's do the punit for it. In this case, you will have a 1:1 ratio of dominant to recessive. So you will have, 1:1 ratio where you will have a black is to white, 1:1 ratio. So basically in a given cross where a dominant has been crossed with a recessive parent, you get a 1:1 ratio of both dominant and recessive characters. Then the dominant parent was heterozygous, and if you only get a heterozygous phenotype, a single phenotype, then they were homozygous, right? So here the correct answer will obviously then be the recessive, completely recessive organism, which is option 4. Moving on to question 119. A pink flower snapdragon plant was crossed with a red flowered snapdragon plant. What type of phenotype is or are expected in the progeny? Now, the moment they say snapdragon and they say pink, the first thing that should strike and come to your mind is the term partial dominance. This is not classic Mendelian dominance. This is partial dominance, right? So now what happens in partial dominance is that if a red flower is crossed with a white flower, instead of getting a dominant red character, you get a partial pink character. You get a mixed phenotype. So in your question, they have said that a pink flower has been crossed with a red snapdragon, right? Again, it's like the test cross, so you will get a 1:1 ratio of pink to white, right? So here red flowers as well as pink flowers will be formed. So option 4 will be your correct option. Moving on to question 120, it's another match the following. Let's see. 2 or more alternative forms of a gene. We know this is an allele. So A has to be 3. Let's look for options with A3. There is only one, option 1. And our question is solved. We know that option 1 is the correct option. We don't have to look further at all. So very easy to solve, right? So for 120, option 1 is the correct option. Okay, going on to question 121. Lecithin, a small molecular weight organic compound found in living tissues, is an example of, this is a phospholipid. It's a straight question from biomolecules. This is a phospholipid. Okay. Question 122, another match the following. Let's see if we can use the same trick here. So when I look at the first list, the 2 of them that strike to me immediately are Saccharomyces cerevisiae, which I know is yeast, and Streptococcus, which I know produces streptokinase. So we will look for B1, D3 options. Sorry, B1, D2 options. Now there's 2 of them with B1, D2, option 1 and option 2. So which means we have to move on to the next best, which for me is Clostridium butyricum. Now that will obviously give butyric acid. So A3, B1, D2 is something that I'm confident about, and there is one option holding that, which is option 1. Okay, which leaves C24, which is Trichoderma, gives cyclosporin A. Okay, so then the correct option for this one will be option 1. Moving on to question 123. In the given figure, which component has a thin outer wall, highly thickened inner walls? We know this is the structure of a stomata, and the guard cells have this particular structure that they are talking about. In the guard cells, they have a thick and thin differentiation in the wall, where in the guard cells you have a thicker outer wall and a thinner inner wall. What it helps in is that during the daytime, when endosmosis happens, when water enters into the guard cells become turgid. And when they become turgid, the cells extend and become more curved, therefore opening up the space for the stomata. So the stomatal pore becomes larger. So obviously the guard cells will be doing it, and the guard cells are marked at C. So C is the correct option, which is option 3. Moving on to 124. Which of the following is an example of an actinomorphic flower? These are one of such questions which, if you know the answer, you know the answer. If you don't, you don't. Right? So actinomorphic amongst all of them would be Datura. Yeah, Datura is an actinomorphic flower. Question 125. A transcription unit in DNA is defined primarily by the 3 regions in DNA with and which are, right, in respect to upstream and downstream end. So a transcription unit consists of a promoter gene, which helps in binding of the RNA pol, then the structural gene, which will express itself, and then finally a terminator in which the RNA pol is going to detach from the segment, right? So that is the correct order, and that is option 2. Promoter, structural gene, terminator. Now, question 126. What is the fate of piece of DNA carrying only one gene of interest which is transferred into an alien organism? So, what happens to your gene of interest after the vector has inserted it into the organism that in which you want it to be, into your host? Okay, so A, the piece of DNA would be able to multiply itself independently in the progeny cells of the organism. So, if you think about what happens to the GOI after it is inserted inside the vector carries it to the competent host. Inside the competent host body, it gets incorporated in the host DNA. And therefore, whenever the host DNA will replicate, transcribe, translate, it will also replicate, transcribe, and translate with the host, right? But if it is not incorporated into the host DNA, if it is free, then it will be digested by the host system. So no, it cannot independently do anything. So option A is obviously wrong. Option B, it may get generated into the genome of the recipient. Yes, that may happen, and it happens, right? So B is correct. It may multiply and be inherited along with the host DNA. Yes, it can. So B and C are correct. We have an option with B and C, and that is only option 1. So this has to be our correct option. We will not even read D and E further. Moving on to 127. Oxen is used by gardeners to prepare wheat-free lawns, but no damage is caused as it does not affect mature monocotyledonous plants. Again, this is a question in which if you know the answer, you know the answer. So, yes, option 1 is the correct answer. Question 128. Cofactor of enzyme carboxypeptidase is zinc. Zinc is the correct option, so that's option 3. Question 129. The lactose present in the growth medium of bacteria is transported to the cell by the action of. So we know there are 3 genes, right? So you have 3 basic proteins. One of them is beta galactosidase. You have permease and you have transacylase. These are the 3 that come out of the lac operon or the Jacob-Monod operon. So now this digests and this permeates, as the name suggests. So internalization happens with the help of permease. So for 129, the correct option is option 1, permease. Let's do question 130. Which of the following can be explained on the basis of Mendel's law of dominance? Out of the one pair of factors is dominant and the other is recessive. Correct, right? So according to Mendel, what he said was, he called our alleles as factors, right? So for tall, you had capital T and small t alleles. Capital T being the dominant one, small t being the recessive one. So this is correct. Alleles do not show any expression and both the characters appear as such in F2. No. In F2, yes, you get both the tall and the dwarf, but you also get a heterozygous character, right? So B is wrong. So A is correct. Let's see. The ones that contain B, let's first do that. Let's do process of elimination. So option 1, 3 and 2, all of them carry option B. So then option 4 is the only correct one left. So we have to go with option 4, right? So A, C, D, E are the correct factors. Moving on to question 131. There are 2 statements. This is not really assertion reason, but 2 statement based questions. So let's do the first one. Bt toxins are insect group specific and coded by gene CRY1AC. This is true. Bt stands for the term, this is question number 131. So Bt stands for Bacillus thuringiensis. They have a protein known as the Cry protein, which is a short form for crystalline protein. In the in the bacteria, it is inactivated, but in the acidic pH of the gut of the insect, it turns into its crystalline form and is capable of perforating the gut of the insect and thereby killing it. This is the technology which has been used in our GMOs, genetically modified organisms, like our Bt cotton, our Bt brinjal, our Bt tobacco, so on and so forth, right? So the first statement is obviously correct. The second statement says that it exists as an inactive prototoxin in the B. thuringiensis. However, after ingestion, the inactive protein gets converted into active form due to acidic pH of insect gut. This is true, and therefore both the statements are true. Option 3 is the correct option. Question 132. Okay. There are two statements. Parenchyma is living but collenchyma is dead. This is wrong because parenchyma is living, collenchyma is also living, sclerenchyma is dead. And gymnosperms lack xylem vessels, but xylem vessels is the presence is characteristic in angiosperms. This is correct. Gymnosperms are vascular. They do have xylem, but they only have xylem tracheids and xylem parenchyma. They do not have xylem vessels. And therefore, the correct option then will be option 2, which is statement 1 is false, statement 2 is correct. Moving on to question 133. Chromosomes become gradually visible under light microscope during leptotin stage, correct? The beginning of diplotin stage is recognized by dissolution of synaptonymal complex, also correct. So option 3, both statement 1 and statement 2 are correct. Okay. Moving on to question 134. Match the following. Let's see. Nucleolus. It is the site for active ribosomal rRNA, ribosomal RNA synthesis. This should be correct. So A should be 3. Now option 1 and 3 say A is 3. So let's go with B. Centriole. Centriole organization like the cartwheel is true for the centriole. So B should be 2, which is option 3, A3, B2, C4, B1. That is your correct option. Question 135. List of endangered species was released by obviously the International Union for Conservation of Nature and Natural Resources. That's your IUCN. Option 2 is the correct option. Now we will move on to section D, which is the non-compulsory section. Out of 15, you had to do 10 in the NEET. We will, however, solve all 15 questions, right? So, question 136. The DNA present in chloroplast. So, chloroplast and mitochondria, as we know, are semi-autonomous organelles which have their own DNA. But their DNA is obviously circular, but it is dsDNA, double-stranded DNA. So, option 4, circular double-stranded, is the correct option. Moving on to question 137. Which of the following is fused in somatic hybridization involving two varieties of plants? Obviously, it's somatic hybridization, so we will fuse protoplasm. Question 138. Identify the correct description about the given figure. Okay, this figure, if you look at it, you can see the stamen flowing out, right? So it should be pollinated in a way where the stamen needs to be flowing so that the anther is easily accessible, and therefore it should be wind pollinated, right? Let's look at the options. So option 3 says wind pollinated plant inflorescence showing flowers with well exposed stamens. So that's your correct option. Going to 139. Spraying sugarcane top with which the following plant growth regulators increases the length of the stem. Now, this is an interesting question because for this you have to, if you remember the discovery of a particular plant growth regulator, you would have been able to answer this question without any problem. And that particular regulator obviously is gibberellin or gibberellic acid. So what happened was that in Japan, rice seedlings were having a disease which was known as the Botany disease or the foolish seedling disease. In the foolish seedling disease, what happened was, instead of giving out branches, a rice seedling would grow tall with very few branching. This reduced harvest, obviously, and therefore everybody started to figure out what was going on when they figured that this particular seedling, this particular batch had been infected with a fungus known as Gibberella fujikori. What this fungus did was that it secreted something known as the gibberellic acid. GA3 was the first gibberellic acid that was discovered, which increases the space between the nodes. So this is a node, this is a node, and in between you have an internode. So it increases the length of the internodes, and therefore plants grow in height. This was discovered and now it is extensively used in dwarf plants to increase their height, and therefore this answer's option would, correct option would obviously be gibberellin, which is option 4. Moving on to question 140. Oh, these are the names of scientists, right? So Griffith should immediately remind you of the transforming principle. What Griffith did was that he took the pneumonia bacteria and he took its two strains, R, which is non-virulent strain, and S, which is the virulent strain. S was killing mouse, R was keeping them alive when injected in mice specimen. Now, what he did was that he took heat killed S and he saw that the mouse survived, right? Which means upon killing it with heat, whatever was causing the virulence is not being able to act on it, right? But when he put them together and injected the concoction, they found that the mouse would die. So he said that, okay, the factor for virulence is somehow getting transferred to the R strain and is changing the R strain into a living virulent factor, right? So it, because it transformed the R strain, he called it the transforming principle. And the experiment was known as the experiment on transformation. So Griffith, which is option A, goes with third in the list too. So A3 should be correct. So 3 and 4, both of them could be correct options. Let's go with Jacob Monod. Jacob Monod discovered the lac operon. So B would be 4. So option 4 is the correct option, right? Moving on to question 141. Again, a match the following. So let's see which one are easy. GLUT4 insulin, very easy. Insulin is an enzyme. GLUT4 is what enables glucose transport into the cell. So A should be 4 and B should be 1. So option 3 is the correct option, right? Moving on to question 142. In C3 plants, some O2 binds to rubisco, hence CO2 fixation is decreased. True, because they do photorespiration, right? So statement 1 is true. In C4 plants, mesophyll cells show very little photorespiration. Bundle sheaths do not show photorespiration. This is false, right? So therefore, option 1 is the correct option. Moving on. Identify the step in the tricarboxylic acid cycle which does not involve oxidation. Again, one of such questions which if you know, you know. So, succinyl-CoA changing into succinic acid is that step. Option 1 is our correct option. 144, another match the following, okay. So citric acid cycle, where it happens, I think should be it happens in the mitochondrial matrix, right? So A should be 2. There's only one option with A2, which is 4. So 4 is your correct option. Now, next is 145. Which of the following statement is correct regarding the process of replication in E. coli? Let's read the first one. The DNA-dependent DNA polymerase catalyzes polymerase in a 5 to 3 as well as 3 to 5 direction. This is wrong. It only does it in the 5 to 3 direction, right? So, the second option, catalyzes in the 5 prime to 3 prime direction, is the correct option. The second option is the correct option. Moving on to question 146. So, in an ecosystem, if the net primary productivity of a firm first trophic level is 100x kilocalorie per meter square per year, what would be the gross primary productivity of the third trophic level of the same ecosystem? Okay, so obviously we will do the 10% rule, right? So for the first trophic level, they have said it's 100x is the NPP. Now we know the gross primary productivity would be 10 times of it. So the GPP should be 1000x in the first trophic level. So if I were to draw the trophic levels, let's say like this, first, second, we need up to 3, right? So if it is 1000x here, then it is 100x here, because only 10% will pass, and then 10% of that would be 10x. So option 1 is the correct option, where 10x is the GPP for the third trophic level. Okay. Moving on to question 147. Okay. Rose is a perigynous flower. Okay. So A should be 2. P has a marginal placentation. So B should be 4. Cotton has twisted estivation, and mango is a drupe. So the correct option would be 3, A2, B4, C1, D3. Another question, even though match the following, you will not be able to use much of logic. You will have to know. Okay. Moving on. Robert May. Robert May is known for the global species diversification, right? So he was the one who said that we have 7 million known global species diversity. So A is 3. Humboldt gave us the species area relationship. So B is 1. Paul Ehrlich did the rivet popper hypothesis, where he said that if you keep on taking rivets out of a aeroplane, initially it will make no difference, but one day will come that a crucial rivet will come out and the whole plane will fall apart. This is what we are doing to our ecosystem. We are killing organisms, leading them to extinction without still facing a very dire consequence, but one day will come when such consequences will occur. This is known as the rivet popper hypothesis, right? And David Tillman gave us the long-term ecosystem experiment using outdoor plots, right? So the correct option is A3, B1, C4, D2. So that's option 4. Moving on to 149, another match the following, talks about adelphi. So now what is adelphi? Adelphi is clustering of all the stamens together. So adelphi is all stamen fused but anthers free. If this happens in one group, then obviously it's monoadelphi. If it happens in 2 groups, then it's diadelphi. If it happens in multiple groups, then it's polyadelphi. Another term that you have here is epiphyllous, which means attached to leaf. Okay, so the stamen would be attached to the androecium, would be attached to the leaf. Now let's see. You have monoadelphi. Monoadelphi is found in China rose. So A should be 4, and that's the 4th option. So for question 149, Fourth option is correct. Sorry, no. 3 and 4 both have A4, right? So we'll have to do B, diadelphys. Diadelphys is found in P. Okay, so then B should be 2. Yes, and then option 3, A4, B2 is the correct option. Option 3. Moving on to question 150. Statement-based questions. Correct statements about brown algae or Phycophyceae. Asexual reproduction occurs usually by biflagellate zoospores. This is correct. Sexual reproduction is by oogamous method only. This is also, sorry, it's not only oogamous, it can also be by spores, and therefore this is wrong. Stored food is in the form of mannitol or laminarin. This is true. Major pigments are found are chlorophyll A, C, carotenoid, xanthophyll. True. So A, C, D are true. You have, and B is false, right? So option 1 is the correct option because all the others have B in it anyway. So this was the botany paper. I hope the explanations were clear. If you have doubts, you can let me know. Thank you.